All right. Well, welcome everyone, and hello, and welcome to the third Dean's Digital Cafe of Spring 2025. I'm glad you're able to join us today. I'm your host, William Letzu, Assistant Professor in the Department of Biological and Chemical Sciences at New York Tech. And on behalf of the CAS Dean, Dean Chuck Moore, I'd like to thank all New York Tech students, faculty, staff, alumni, and guests here for joining on to today's event. So I'd like to remind everyone that today's talk is being recorded. We'll open the floor for discussion at the end of the presentation, but you should feel free to enter questions or comments into the chat at any time. And also stay tuned for our new Dean's Digital Cafe web page on the CAS section of the New York Tech website, where you'll be able to watch recordings of today's and past events. So that's coming soon, hopefully. Before introducing Dr. Ruiz, I'd like to acknowledge the land. The Long Island campus of New York Tech sits on a larger territory known as Pominoke, which was home to numerous indigenous communities, including the local tribes of the Matcocks and the Merricks. I'd like to acknowledge the land, the stewardship and sovereignty of the Native peoples, and their enduring importance, even in this virtual environment, to our work and community here today. And now it's my great pleasure to introduce today's speaker, Dr. Henry Ruiz. Dr. Ruiz joined New York Tech as an assistant professor in the Department of Biological and Chemical Sciences in the fall of 2024. Before coming to NYIT, Dr. Ruiz earned his doctorate in neuropsychology from the Graduate Center at the City University of New York, where he studied the role of neuropeptides in inflammatory bowel disease and its associated pain behaviors. There he developed expertise in rodent metabolic phenotyping and further investigated the pathological role of advanced glycation end products, or AGEs, in metabolic diseases during his postdoctoral training at Mount Sinai and NYU Langone. As a research assistant professor in the Department of Medicine at NYU, Dr. Ruiz led NIH-funded projects aimed at uncovering the mechanisms by which AGEs contribute to insulin resistance and obesity. His current work at NYIT focuses on an NIH-funded project to identify the cellular mechanisms that promote lipid accumulation in adipocytes or fat cells, with the aim of discovering potential therapeutic targets for the treatment of obesity and other lipid-associated cardiovascular disease. Dr. Ruiz's long-term goal is to integrate his training in neuroscience and metabolism to study the central nervous system regulation of glucose and lipid metabolism, particularly in the context of neurodegenerative diseases such as Alzheimer's. Today, he will be telling us about his exciting new work on the surprising role of a cytoskeletal protein called Diaphanous 1 in lipid metabolism. So welcome, Henry. The floor is yours. We're excited to learn more.
Thank you, William, and thank you to everybody who was present. This is very exciting, and thank you for the very kind introduction. All right, so I'll tell you guys today a little bit about the research that I do. As William indicated, I've been at NYIT since last fall, so my lab is still in the process of developing, but we are up and running. So I'll tell you a little bit about the background research that I've done while at NYU. So my research, the one that I'm choosing to present today, is centered around obesity, right? And obesity, we all talk about it, everybody talks about it, and sometimes we don't even have a definition. So typically and clinically, obesity is defined as a body mass index, which is based on your height and your body weight, essentially your body weight in kilograms and your height in meters square. Right, so if that ratio comes up to a number of 30 or higher, then you're considered to be obese. Now, most of us at one point or another have seen a chart like this, right, when we go to the doctor, and then we quickly try to find ourselves, where do we fall in terms of the height, where do we fall in terms of the weight, and where in this BMI ratio do we find ourselves. And as you can see, essentially anything above 30 or anything that is in the orange and red is considered to be obesity. Now, obesity, there are multiple levels of obesity because of this continuous form in which we characterize it, right? So there is a normal weight, there is overweight, and then there are three different classes of obesity depending on how much adipose or how much adiposity, which is just a fancy term for how much fat we carry, right? Now in America, so the CDC has been very good at keeping a series of records with regards to how much, how prevalent is obesity in the United States. So here I'm showing you the CDC charts from 2011, right? So 14 years ago. What I want you to appreciate from this graph is that in 2011, again, about 14 years ago, if most of the states fell in this green-yellow color, which indicates that their incidence or the prevalence of obesity was relatively low, right? Below 20% in most states, or below 30% in most states. Now, as I walk you through the years, so here, this is the map for 2012. This is the map for 2013. This is that same map for 2014, for 2015. And what I hope you guys are appreciating is a continuous change towards the darker colors, right? 2016, 2017. And now we're starting to jump into only some states having prevalences under 30, and then the majority of the states going above 30. And we continue 2019, 2020. Now we see that we're getting even darker colors, 2021, 2022, and 2023. Essentially, we are now all in the red, right? So as of now, 2 years ago, 1 in every 5 adults in the United States is obese, right? So this is a clinically relevant problem. We have a problem of obesity, and it's a problem that is also now not just limited to the United States, but is moving everywhere else. Now, recent advances in pharmacology, right? So you guys are probably very familiar with the Ozempics and all the different pharmacologies that were developed to treat diabetes and insulin resistance, but we found that they're very good at suppressing your appetite, right? So they make you not want to eat. And that's a good thing, because we get people losing 25% of their body weight. So we are now being able to bring in these numbers. However, I think that's an artificial way of tackling the disorder, right? It is unbelievable in my mind that after many years of research in terms of understanding adiposity and how we become obese, that we really don't know what are the cellular changes at the level of the fat cell that are promoting obesity. And that's essentially where I want to focus my research, or one of the legs of my research. So adiposity is at the core of who we are, but it's also at the core of obesity, right? So we have the fat is there for a reason, right? So think about it evolutionarily, when we were hunters and gatherers, when we were running after the lion and after those animals that we needed to hunt, we needed to be able to not only collect the food, but also store it in our system. And I think this is how adiposity or these fat storages developed throughout evolution, right? So there are places where we store excess nutrients. Now, this is all good, and this system is very advantageous when you are in a nutrient-deprived environment, such as those hunters and gatherers, right? Now, with all the advent of all this ultra-processed food and the high availability of foods all around, it is when it becomes a problem, right? Because now we have all these organs that are there to store the fats, to store the nutrients, but we have so much nutrients coming into our bodies that it now becomes a problem, and it leads to obesity. Now, the one thing about fat, so we have 2 primary types of fat in our bodies. This is mammals and in humans. We have what is known as white adipose tissue, right, which is what we see here in the yellow colors. And that is the fat that typically you will find under the skin, around the organs, and in our bellies. And that's what allows us to store nutrients long term, right? Then we can break them down whenever we need energy, and then turn that into some sort of fuel. There's also another type of fat that is seems to be much more primitive, and we are in the process of losing it, right? So, and that is called the brown adipose tissue, or BAT. And it's called brown adipose tissue because of the color. It's really a brown color. And what gives it that brown color is the high mitochondria content, right? So if you don't know anything about mitochondria, I'll tell you mitochondria, people typically summarize it as the power house of the cell, right? Is the one that is in charge of breaking things and turning them into energy. So this brown fat contains lots and lots of mitochondria. So it's very metabolically active, right? Now, when you turn obese, when a person turns obese, we tend to lose that brown fat. And then the white fat tends to expand, right? It's storing all that fat. And then we tend to find fat in the liver, find fat in our muscles, in our heart. And that's where it becomes a pathology or a problem to our health, right? Now the question is, how do we stop that? Or do we understand why, or how can we prevent those adipocytes or the fat cells from engulfing all these extra nutrients? The other thing that happens in obesity is, as we can see here, so these are fat cells or representation of fat cells during in a lean individual. As we become obese, that fat cell is no longer just storing nutrients, it becomes actually inflamed. So all these immune cells, which normally will respond to things like bacteria, viruses, and so on, start to detect this fat, these large fat cells as pathogenic, and they want to get rid of them. And so they infiltrate the fat tissue and then they cause dysfunction. Now the cells are unable to function like they normally would. And that is a problem, right? Now, so this is my representation of a fat cell, right? Here we have a fat cell, and inside of it, it has just a lot of fat. Under normal conditions, our brain, right, via the sympathetic nervous system, which are these fibers that you probably have heard at one point or another, are nerves that are part of the fight or flight response. Those fibers come and innervate the fat tissue, meaning they actually have little ends that go and secrete things directly onto the fat. And those things are called catecholamines, right? Those catecholamines stimulate a specific type of receptors on the cells, right? Receptors are essentially just these molecules that are able to read the code coming from the brain. And then when that gets activated, it initiates this pathway, right? Which is the lipolysis, right? And lipo comes from fat, lysis from breakdown, so it's the breakdown of fat. And as you can see here, activation of those receptors first stimulates this enzyme called ATGL, which begins the breakdown of a triglyceride. So you probably are familiar with a triglyceride because when we go to the doctor, we get a number almost every year. We get a number of where our triglycerides are, and if my assumptions are correct, at least 1 in 5 of us will have had high cholesterol or high triglyceride levels, so we are somewhat familiar. Anyway, what I want you to focus on is the structure. So it's a glycerol backbone with 3 fatty acids, therefore does the triglyceride, right? So there's 3 fatty acids attached to a glycerol backbone. And then when lipolysis is stimulated by our brain saying, hey, go and break those fats into smaller things, ATGL removes the first fatty acid, then HSL removes the second fatty acid, and MGL removes the third fatty acid. Right? And then you end up with a whole bunch of fatty acids running around, and that's not a bad thing because fatty acids are nutrients, they're energy, they can be turned into energy. And the glycerol backbone, right? And then these get secreted, and then our organs, our brown adipose, our liver, our muscle will utilize these fatty acids, turn them into energy, and then you can run or run from that tiger again, right? So this is the normal process. Now, another fate of those fatty acids, or nowadays when we eat those big burgers that are, rich in saturated and unsaturated fatty acids, then another fate for fatty acids is that mitochondria. Remember that powerhouse I told you about before? Well, every cell has it. Fat cells have it too. And what can happen is that those fatty acids can be brought inside the cell and then directly utilized by the mitochondria in the cells, right? So in one way, in lipolysis, we're just taking a triglyceride and breaking it into its parts. And then fatty acid oxidation is taking the actual fatty acids that can come from diet or can come from lipolysis, right, as we see here, and then feed them into this little powerhouse that will turn them into energy, right? Those are the two potential fates for fatty acids. Now, some research that was published in 2019 from the work that I was doing at NYU suggested that there is this protein called the receptor for advanced glycation end products, or RAGE, right? And RAGE, we show that it probably developed while we were hunter and gatherers, right? because its job is to prevent the fat cell from breaking down triglycerides, right? So it prevents lipolysis or the breakdown of fats. So, it was a nice story. It was novel. We showed it, and we showed that if you remove RAGE, right, and therefore you allow now the adipocyte to break down the fats, then those animals do not become obese, and they are instead they use their energy to generate heat, something known as thermogenesis, right? You're generating heat, utilizing those fatty acids for heat instead of storing them as fat. Now that was nice, and then eventually we realized, or we found, we brought into perspective the fact that RAGE and that receptor that we took out in those animals in this paper, it talks with another protein called diaphanous 1 inside the cell. So then the question that came up was, do we need this protein? Like, does this protein, diaphanous 1, play any role in promoting obesity? And what was known about diaphanous 1 is essentially that it's an actin polymerization protein. That's just a fancy term for saying that it regulates so these tubules are formed inside of cells called actin fibers, right? Actin fibers play a role in many things, from cell division to the reorganization of cells, right? Especially if you're a fat cell and you need to store more fat, then you need to be able to be flexible and increase in size and so on. So we thought that this is what was happening, right? We said, okay, maybe diaphanous 1 regulates the size of the adipocyte, right? Well, we looked in the literature with the help of some bioinformaticians at NYU. We looked in human fat, and we looked in rodent fat. So rodent is a very commonly used model, specifically mice, for obesity, because we can feed them a diet that contains a lot of fat and they become obese. And they show many of the symptoms that obese humans do, such as a lack of response to insulin and the inability to break down sugars such as glucose. So when we look at those tissues, at those fat samples, we look for the expression of this protein DIAF1. And here, the darker colors, the redder colors indicate that the expression is elevated, and the lower colors, the cooler colors indicates that the expression is suppressed, right? As you can see from this graph, almost all the individuals, human individuals in the fat that were insulin resistant, right? So those that will likely develop type 2 diabetes, versus those that were sensitive to insulin, so they were more responsive to insulin, and those tended to express more of the more of diaphanous 1 in their fat than the healthier counterparts. Now, this was not ubiquitous, right? There was that one study where this association didn't show, but overall, I think we can say that high levels of this protein in fat tend to associate with obesity and obesity-related metabolic impairments in humans, and in the rodents, it is very strong association, right? The darker red colors say, yes, there is more of this protein in the fat from the rodents that are fat compared to the rodents that are not fat. And interestingly, there was a study in rodents where they made the mice fat and then they made them lose weight. So now in those animals that lost weight versus the ones that didn't, they actually find the diaphanous 1 expression is lower. So the opposite, right? The fat mice have more, and then those mice that lose weight have less. So altogether, this gave us a good grounds to study whether, does this protein play a role in regulating obesity? So I found a set of collaborators at the University of Virginia, and who had access to human fat samples from obese and very obese individuals. We're talking BMIs above 40 and 50s, right? So severely obese individuals. And then what I didn't tell you before is that the white adipose tissue, remember there's 2 flavors, white and brown, but within the white flavor, there is different types, and it's usually depending on the location, right? So we have subcutaneous, which means right under the skin, and we have omental, which is in the omental cavity in our bellies, right? I was able to get my hands on that fat and those very precious fat samples, and I analyzed them for the expression of the genes that encode the proteins that of interest, so such as RAGE, diaphanous 1, and some markers of inflammation, such as TNF, CCL, and CD68. And then what we found in that paper, and we published this work in 2021, I believe, is that while in those severely obese individuals, the amount of DIAF that was expressed doesn't really associate with obesity, right? Because again, we're talking about very, very extremes of obese individuals. The amount of DIAF in the fat was very predictive of how much insulin resistance those individuals had, right? The propensity to develop type 2 diabetes. So the more protein, more of this diaphanous one that you have, the greater the chances of you developing or having insulin resistance. So I think that together, you know, these studies gave us a good grounds to study this in animals. So we move now to animals, and the very first question is, okay, we're going to knock down or genetically remove diaphanous 1 from these mice. But remember that DIF1 plays a key role in the regulation of actin dynamics. So the question was, if we remove it, do we mess up with the adipocyte, the fat cell's ability to rearrange, right, to use those actin fibers? And the answer is essentially no. So this is an actin polymerization assay, which essentially what it does is it looks at the cell's ability to put actin fragments into the fibers, right? Are they able to elongate, essentially? And you can see that they're right on top of each other, the cells that don't have DIF and the cells that do have diaphanous. So diaphanous 1 is not necessary for adipocyte actin polymerization, right? So we got that out of the way. We did this in primary adipocytes that were directly derived from mice. So then we went ahead and generated a mouse, right? A mouse that completely lacks diaphanous 1 specifically in adipose tissue. So we use there's a specific technique, a genetic technique called the Cre-Lox recombinase technique that allows you to target the deletion of a gene to specific organs, right? So in this case, we use adiponectin as the promoter. So it removes the gene of interest specifically from cells that express adiponectin, which happen to be only fat cells. When we do that and we put the animals on a high-fat diet, right, to make them obese, we see that here the blue lines, and this will be consistent throughout the presentation, blue lines are the normal, quote-unquote, mice or wild-type mice, and the reds are the mice that do not have diaphanous 1 in their fat. And you can see that the wild-type mice gain weight across, I can't recall right now, 12 weeks of high-fat feeding, and you can see that there is not much, that there is a significant weight gain, right? So they go from about 22 grams to nearly 40 grams of weight. They almost double their body weight. That doesn't happen to the same extent in animals that do not have DIF in their adipose tissue, right? They still gain some weight because they're still growing, right? And you're still getting excess nutrients, but it's not to the extent. So when we do a DEXA scan, which is essentially a radiographic scan that tells us how much of the total body mass is attributed to bone versus fat mass, we see that in terms of lean mass, so muscle, fat, muscle, bone, water weight, and so on, there is a decrease in the knockout animals, in the ones that don't have DIF. But more importantly, in the fat depots, the total fat is much more decreased in the animals that don't have DIF in the adipose tissue. So when we sacrifice these animals and collected the tissues, we measure how much the fat parts weigh, right? The white adipose tissues and then the brown adipose tissues. And as you can see, it's mostly in the white adipose tissue that there is a decrease in the total mass, right? So those animals are not becoming obese because they're not storing the fats in their white adipose tissue. And then when we perform a histology study, essentially taking those fats, cutting them, staining them, and then analyzing, you can see here, this is what a normal cell, fat cell looks like. So this is the brown fat, this is the white fat in subcutaneous or inguinal, and this is the omental or epididymal fat. And you can see that, again, very well defined, nice size adipocytes. But in the wild type animals, when you put them on a high fat diet, all of these adipocytes begin to store the lipids, and then they begin to enlarge and lose their nice shapes. Even the brown adipocytes, you see all these little empty holes, They're just triglycerides that accumulate. So then the question became, why are these animals not packing their adipocytes with fats, right? And is this beneficial, right? So I told you that one of the problems with obesity is that you stop responding to insulin, right? And you stop being able to break down glucose. So the way that we test this in mice is we provide them a bolus or an injection of glucose, and then we measure their glucose levels across time. In you or me, if we have a meal within 2 hours, our glucose will start at about 100 milligrams per deciliter or 90 milligrams per deciliter. And then 2 hours later, they will rise. And then 2 hours later, they'll come back to about the same level, right? Pretty similar things happen in the mouse, right? So they start out about 150. And these are mice that were fed a high-fat diet, so that's why they're a little bit higher. And you can see in a wild-type animal that is obese, you give them glucose or sugars, and then the sugar levels increase, and then they remain elevated for quite some time, right? So you see that even 2 hours later, their levels are still very, very high compared to what they were before the bolus, right? This is the definition of being unable to metabolize glucose, right? This is somewhat, to a large extent, enhanced or protected in the animals that just lack this one gene in their fat tissues, right? So they're better able to clear glucose. Now, it's not a complete protection, right? But it is is a significant effect such that these guys might be in a better position to become metabolically healthy if you remove the fat diet. Now, on the right side, we have an insulin tolerance test. So when we have a meal, our glucose levels rise, our pancreas secretes insulin, insulin goes to all the cells, tells, hey, uptake that glucose and bring it back to normal levels. That's the typical response, right? So glucose levels drop after insulin. This is what's happening in the wild type animals, those fat obese animals. You give them an insulin bolus, and yes, they drop, but their drop is not very drastic, right? So if you see, we go from about 175 milligrams per deciliter to about 100. So it's not a very drastic drop. And then they just remain at that level, and as slowly they come back. If you look at what happens in the animals that don't have diaphanous one in their fat, that drop is significantly lower, and then it remains lower for a longer period of time, right? Which tells us that those mice, just by deleting that one gene in the adipose tissue, now you make them more or better able, rather, to respond to insulin, even when they're being fed these very high-fat diets. So then the question became, okay, why are they leaner? Why are these mice not packing up the calories? Is it that they're utilizing them more? Right, so at NYU, we have these nice little chambers for mice that allow us to measure energy expenditure, right? So how much energy are you expending, right? And this is based on indirect calorimetry using oxygen consumption and CO2 production. And then, using very fancy algorithms, they can estimate, okay, this is the energy expenditure. If you spend more energy, obviously you're going to be or the likelihood of being leaner is greater, right? And most obese, and some people used to think obesity was because we were not spending energy. We were sitting on couches all the time, right? Now, when we analyze these animals, we see that, again, the animals that don't have diaphanous one in their adipose tissue consistently have more energy expenditure, right? So they're utilizing something inside their bodies, utilizing the nutrients much more effectively than those animals that were just obese but were normal. All right, and this is just showing that the regression, just because some people like to see it that way too. So at that point, we reached a level essentially where we knew that these animals are protected. So we knew that of the relevance of diaphanous one in obesity and metabolic dysfunction. We knew that it happens in humans and it happens in rodents. We knew that if we delete it in rodents, we protect those rodents from obesity. And then the question is, okay, but what is happening at the level of the cell? So now we know that if you delete diaphanous one, those cells remain small. But why do they remain small? How come they don't become enlarged like we see in the wild type? So we had to now move to a different model where we can manipulate the cells themselves, right? So I had to come up with a way to mimic the high-fat diet or the burger but on cells that are grown on a dish, right? And well, just using very logistics and the simplest experiment that one can think of is those burgers and those high-fat diets and so on, they're rich in fatty acids, right? So specifically in palmitic and oleic acid. What happens if I just provide this to the cells? So I performed this study where I took my cells, right? I differentiate them. That's just a fancy word for saying that we put some chemicals that will make them turn into fat cells, right? They become fat cells. And then after they're fat cells, so you can see here, they essentially form these little droplets, right? Inside which they store their triglycerides. So each one of this is a cell. So that's one cell, that's one cell, that's one cell. Here's a pretty one, right, with all the little lipids around it, around this nucleus. So those are my adipocytes. If I give them this burger, right, the palmitate and oleate mixture, then those cells become like this. You see now there's more of those little droplets. There's more of those lipids building up. So they're taking those fatty acids that I'm giving them and putting them into a storage mode. And then what I did was I took these cells and I extracted all the lipids and then I quantify how many triglycerides, or I measure how many triglycerides are there. And we see that the treatment with this palmitate and oleate cocktail increases the amount of triglycerides that accumulate inside the cells. Then I took this and I did it, the same study, but I did it in cells that were derived or that came from a wild type animal or an animal that lacks diaphanous 1 in the adipocytes. And then after exposing them to the fatty acids, we can see that in the wild type cells, and here red is staining for neutral lipids, so mostly triglycerides. The green is staining for a protein that usually surrounds lipid droplets, and the blue is staining for DAPI, which is a nuclear stain to separate the individual cells. You can see that the lipid droplets are much bigger in the wild type cells than they are in the cells that derive from the DIAF1 knockout, right? When I extracted the lipids and quantified the triglycerides, I can find that indeed these guys store less of those triglycerides or those fatty acids into triglycerides. All right, so I'm gonna bring you back to this little schematic again, showing you what lipolysis is, and then showing you what lipolysis is. So if we want to measure lipolysis either in a mouse or in cells, what we normally do is we starve the cells or the animals for a couple of hours, nothing that will harm the animal. And then we measure how much glycerol and how many non-sterified fatty acids are released, right? Because these are the byproducts of lipolysis. When we did this in the cells, we see, and here the ones that do not have DIAF1 are the red ones. The ones that do have DIAF1 are the blue ones. And this is just a genetic manipulation that I use to knock down DIAF1. We see that those cells that do not have DIAF1 have more non-sterified fatty acids. They have more glycerol, right? Even a baseline. So this is here, vehicle is the baseline. So just after you starve for 2 hours, and then CL316243 is essentially it's a pharmacological agent that mimics catecholamine, so it stimulates lipolysis, right? When we give that drug, we see that the cells that do not have DIAF, they respond significantly greater. The lipolytic response is greater than in the wild type cells, especially if we look at the non-sterified fatty acids. So that gave us a very good evidence that these cells are not storing as many lipids because they have more lipolysis. They're constantly breaking down the fats that they're sterifying from the diet, right? So they take in the fatty acids from the diet and then they burn them in using lipolysis. Now the problem is that, as I told you, when you do lipolysis, you end up with very, very high levels of fatty acids. And fatty acids must be either utilized or they will be turned into triglycerides. So when fatty acids, I showed you before, when fatty acids get used by a cell, it's in the form of fatty acid oxidation, right, by mitochondria. So what we did was we used this very nice little tool that we had at NYU called the Recipher, which is you have your cells in a plate, and then it's a sensor that goes over the cells, and it quantifies or it measures how much oxygen those cells are taking up, right? If you remember your biochemistry, you remember that when you do fatty acid oxidation, one of the byproducts of fatty acid oxidation is oxygen, right? So you need to consume oxygen, and then you will release CO2. This is what's happened. So what we utilize that knowledge and the existence of the system at NYU to test whether if I provide fatty acids, are those cells increasing the amount of fatty acid oxidation that they're doing? So we had cells, and you see that before I add the fatty acid treatment, they are about their respiration is about the same between the cells that have DIF and the cells that do not have DIF, right? So what that tells me is there is no problem at baseline, but when you introduce a stressor such as a high fat diet, or in this case the high content of fatty acids, then the cells that do not have DIF, you can see both cells increase their respiration in response to the fatty acids, but then the wild type cells, they eventually stop responding. They stop oxidizing those fats, and we know that they were putting them into triglycerides, right? So instead of having them be burned, they're being stored. Whereas in the other cells, the ones that do not have DIF, there is a continuously high rate of burning down those fatty acids by fatty acid oxidation, right? And you can see that this is almost 100 hours. So this is a few days, which in cell talk is a long time, right? And you can see that they sustain this increase in fatty acid oxidation. So not only are they breaking down the lipids, the triglycerides that they're forming, they're also burning the byproducts, which are the fatty acids that result from that. So these two mechanisms, or these two cellular events, are significantly enhanced when we remove diaphanous 1 from the adipocyte. You get more lipolysis, therefore you get less triglycerides forming, and then those excess fatty acids go in and feed another pathway, which is a mitochondria pathway, which generates energy and heat and so on. So the big take-home message for us is that we are able to demonstrate that if you delete this one protein, or rather this one gene, from the adipose tissue of rodents, which is elevated in obese humans and obese rodents, if we delete it, then we prevent obesity from developing, and we enhance insulin resistance, right? Now we know that the reason why that is because when you take this protein away from the adipocyte goes away from the storage state where it just wants to store all the fats to a more metabolically active state where it is now breaking down the lipids and utilizing them at the same time. And so this is where we are now. Now that I'm here at NYIT, my goal is to continue to do this work, right? To continue to finish this and be able to publish it. But some of the RNA sequencing studies that we did from these same animals gave us some hints about other proteins that might be regulating how fat cells become enlarged, right? So one such protein is a zinc finger protein 949, which is the center of an NIH grant that I currently am trying to transfer from NYU to NYIT, which would allow me to study what is the role of that protein in mediating this enlargement of fatty acid of adipocytes. So that's one. I'm still open to collaborations. It is very nice to be able to do all this work in mice, but if we can move this to humans, and I know here at NYIT we have a DEXA scanner. We have patients, so I'm looking forward to having the opportunity to collaborate with other people at the COM, whether it be in a human study or, local universities and local medical places to get access to human obese fat tissue and lean fat tissue. And then just in general, whoever is interested, I love science and I look forward to being able to collaborate with fellow scientists. That's why I'm very thankful that William is leading this effort of the Dean's Cafe, because it's a great opportunity to see what everybody else is doing. And this is my acknowledgement page, acknowledging not only the people that contributed to the work that was presented today, but also to the financial supporters and of course now NYIT, who's housing my lab. And I'll be happy to try to answer any questions you guys might have.
All right, thanks so much, Henry. I've had a great talk. Do you have any questions for the audience, from the audience?
I have a question.
Talk to me,
Christina.
Christina.
Hi, Henry. I'm just curious, and I don't have a scientific background, but did you notice or haven't you done any studies about Diaphanous 1, like the effects of that protein on like the feeding timings? Like, have you looked at it in relation to like an intermittent fasting diet where, because I know that there's a lot of talk about that of helping with insulin resistance. So I don't know, like, how do you regulate that particular protein if your studies are showing that not having that protein helps with things like insulin resistance and obesity, then what's sort of like the takeaway from like, okay, well, how do you prevent the accumulation of that, I guess?
Christina, that's an amazing question. It's very, very thought provoking. So I haven't thought about it in the context of intermittent fasting, but I've thought about it in the context of weight loss, right? So we know that we become obese, and then if we restrict our caloric intake, then we will lose weight, right? Now we can't keep it off, but we will lose it. My question was, what happens to Diaphanous 1 during that process? But your question is also it's also a very good one, right? We have models in mice where we can do intermittent fasting, and to be able to tell whether the changes at the level of the protein will happen that quickly will be amazing. So it's a great scientific question. I do not have the answer to, but thank you.
Yeah. Hi, Henry. This is an excellent talk. Can you hear me?
Yes, Hisham.
Yeah. So, are there any mutations that are known in humans of that gene? And is there any, if they exist, is there any link between these mutations and overweight or obesity or anything like that?
Hisham, another amazing question. So yes, there is a mutation on the Diaphanous 1 gene in humans that has been identified by GWAS studies, as gene-wide association studies, right? Where they take all the genome of humans and then they just look at what genes are changed. So some have been identified. Actually, I'm pretty sure it is a Middle Eastern population, but it's not associated with obesity or metabolic syndrome, but actually with deafness, right? So we know that Diaphanous 1 also plays a key role in hair cells that mediate hearing. Now, it would have been ideal if in those same studies they would have looked at the body weights or body composition of those patients, but we haven't published this work. It's hopefully in the last stages of publishing. But this would have been very informative in that study if they would have done, body composition, BMI, and so on. So I do not know the answer to that yet, but we do hope to revisit those published GWAS studies eventually.
Okay, I'm sorry. One more question quickly. Since this is a bad protein, obviously, at least in the context of overweight, it doesn't seem that basally it does do much, because you didn't really present the data about basally. So it seems that you will only need to stress this mice, for example, like the high fat diet and all of that. So are there any drugs or are there even in general any factors that are known to down-regulate that protein so that you could use that to modulate the obesity? Is there anything known about that?
Yes, so there is nothing clinically being used that we can say we could repurpose. Remember that this is a cytosolic protein, so the drug has to be able to get inside the cell. So there isn't much. My previous mentor at NYU is currently developing a drug that will prevent the communication between RAGE and diaphanous. And they're commercially available drugs that will target DIAF, but not specifically. So no, there's nothing current that will allow us to test just selectively inhibiting that.
You will find that out.
Yeah.
Thank you.
Michael has a question.
Hey, Henry, great presentation. Just a quick question. When you looked at this, obviously, you know, free fatty acids are not the only indicators of lipid metabolism. Have you looked at other ones like LDLs and HDLs and cholesterol levels in these mice?
Yes. So thank you, Michael. Yes, cholesterol. So LDL, HDL, and all those guys that are more typically, obesity research stays away from them because you don't want to get the atherosclerosis people mad. But we did measure them. Cholesterol is also significantly lower in the diaphanous 1 knockout animals. So it tends to be a general lipid. So Anne-Marie did some work on diaphanous 1 in the liver, and then she showed that if you knock it down specifically in the liver, then you prevent the high levels of cholesterol. But even in our case, in this case where we just deleted it in adipose tissue, those mice also show decreased levels of cholesterol.
Thank you, Henry.
Henry, I had a question as well. So I really liked at the beginning how you brought in like the evolutionary perspective, how like we needed to store fat when we were in a food scarce environment. So do you think, and in contrast that, we have these fine-tuned biological systems that are there for a reason. So do you think that the reason like we're having these like problems with obesity is not because it's like natural, it's not beneficial, it's just that it was never under selection. There was never a time period where there was food surplus that we have now, so that these genes were just never optimized. So like this in tweaking the system, you're not really causing any problems. You're just, you're touching something you shouldn't touch, but that you're putting it under selective pressure that it hasn't been exposed to previously.
100%. So and thank you, Will. So yeah, so I agree with, I think right now we are living the selection stage, right? So I think that evolutionarily, again, those of us who have that gene and who are predisposed and then make it worse by eating all this excess food and becoming obese, we are going to be phased out. And so I think it has been, if we look at it, if we continue with the idea of an evolutionary development, I think that yes, we're still going through that process, and eventually DIAF1 and RAGE will be kicked out of our fat cells because of the environment that we live now, where foods are so readily available.
Yeah, because I mean it gets back to like what Hisham was mentioning about it with the drugs, like if it has off-target effects, like it's not necessarily bad to target this protein, right? We don't like, it might not even be useful, right? Because it's just wasn't ever selected. So I think that it might be more chance of a success in that sense.
So sorry again, Henry. So what do you think if you started these mice? You did the high-fat diet, overfeeding them. So what do you think if you starved them?
So what do I think if I starve them to induce weight loss or just a regular starvation period?
Yeah, well, it's again, in one way, you try to make them obese. So what will happen if you try to make them lose weight?
Correct. So yeah, so that's something that I wanted to take with me. That's something I want to do here at NYIT is the idea of what happens to this protein during weight loss. And again, Christina gave us another great idea is what happens during intermittent fasting, right? When they are essentially starved for a period of time. I can tell you though that if I give the CL drug, right, which is essentially a catecholamine or a beta-3 receptor agonist, which mimics what happens during starvation, those animals show greater lipolysis, right? So they immediately break down their lipids much more than the wild type animals. So yes.
So they will be more sensitive to losing weight.
Correct.
Right?
That's the hypothesis. Yeah. So that's why I look for if I ever get my hands on those animals again to do weight loss studies about what happens if you remove DIAF. Do you enhance the ability for those animals to lose weight And if you saw that in the cells, this is a sustained effect, right? Is it the same case in the animals? And now just by switching the animals to a regular diet, are we able to sustain the weight loss? Those are questions that still remain open, but they're great questions.
Thank you.
I think we may have one or two students in the audience. Any questions from them as well?
William, I think Ben Albrin has been.
Ben. Okay, I didn't see him. Ben. You have a hand up?
Here we go. Can you hear me?
Just go ahead. Go ahead.
Hi, Henry. Wonderful talk. Thanks so much for that. So welcome to NYIT, and as you know, we're one of the foremost imaging centers and becoming better and better all the time. When I was at Einstein, I had two R01s. The first R01 was imaging actin dynamics in real time, super resolution. I'm sure you can think of ways you can use imaging for your some of your stuff. So what's the plan?
Absolutely. Thank you.
And how can we help?
Yeah, no, absolutely, Ben, and thank you for being here and for the feedback. So now, so I'm really interested to understand what's happening to the fate of the fatty acids, right? Because there is this idea that, if you take in, so in adipocyte, there is the novel lipogenesis, right? So making new triglycerides from nothing, or rather not from nothing, but from glucose, or versus breaking down triglycerides and then making them again from the same substrate, right? So one of the things that I actually proposed in another one that I just submitted last week was to trace, right? Use fluorescently labeled glucose or fluorescently labeled fatty acids that we provide in the diet, and then see where is the fate? How much of this increase in fat cell size comes from you turning sugars into fats? How much is it from just resterification? And I think that would be something that I know that the COM has this little instrument that allows you to trace the movement of individual dots, right? So a glucose dot, where does that glucose dot end up? Does it end up in a triglyceride? Does it end up in another fate? Right? So that's something that I will definitely be looking forward to some help from the COM.
Okay. Great. Any final questions? A great discussion today.
Yeah, well, how about the expression in other tissues, Henry? Again, sorry, it's an interesting talk, so.
Absolutely, no worries. It's ubiquitously expressed, right? So it's anywhere where actin it plays a role, which is almost every cell in the body. So that's why. I mean, if you look at, so I only presented the data from mice where we selectively deleted in adipose, but we also have the global knockout, right? And the global knockout mice, they look just fine. So actin, even though it was described as this actin cytoskeleton protein, it is not crucial for that. But it's expressed in almost every organ, in almost every cell, and deleting it everywhere doesn't make things worse. It actually enhances the protection. Because in those animals, then lipids are not getting stored in the muscle, not in the liver, and so on.
And have they done any work on this global knockout animals and also like high fat diet? And if they did, what do they see?
Complete protection. So you saw that my adipose specific gained some weight. The globals do not gain any weight at all.
So it's more dramatic of a phenotype than the adipocyte specific phenotype.
Correct.
Which means it also regulates energy in other tissues like muscle, which then you may try to combine, for example, a knockout in the muscle and the adipocytes.
Exactly.
And see
or even do the specific knockouts themselves, right? So if I knock it out in muscle, do I still get the protection? And then you can see the relative contributions in each organ. Yeah, no, that's a complete research program there that one can develop.
Yeah, that's interesting.
Right. Well, great talk today. So thank you, Henry, and thank you all once again for joining us today. Please join us again after spring break when Professor Subhabratan Chaudhry, also of the Department of Biological and Chemical Sciences, will be telling us about the development of pyruvate carboxylase inhibitors for studying cancer cell metabolism. So take care, everyone, and have a great rest of your day.
Thank you, everybody. I appreciate the attendance and the feedback.